This is Ganesh, lecturer in zoology, M.R College, Vizianagaram. In our last class, we discussed the incomplete dominance in interaction of genes. Today, we are going to discuss the codominance in interaction of genes, a new concept. The codominance. Let us observe what is codominance. Codominance is a relationship between two versions of a gene. If the alleles are different, means dominant allele and recessive allele. The dominant allele usually will be expressed while the effect of the other allele called recessive, which is masked. All we know that. If the two dominant alleles from different gametes fuse and complement each other to express one character, which is called codominance. Okay, next slide. Okay, let us observe the examples for the codominance. The examples are AB blood group, MN blood group, skin, hair color in cattle, combs in fowls. Okay, now we'll discuss the codominance in blood type AB or blood group, AB blood group. Okay. Here the slide. So the parents are blood type A and blood type B. So the parents homozygous dominant blood type A is represented by IA, and also the homozygous dominant blood type B is represented by IB. So both are dominant alleles. Here IA are dominant alleles and also IB are also dominant alleles. Alleles means genes. So here dominant parents are participating. Then the gametes, this gamete receives IA, dominant gene. Here also the gamete receives the dominant gene, which is IB. So these different gametes with dominant alleles are the genes fuse and form IB. This is a new character or new trait, that is blood type AB. IB represents the blood type AB, homozygous dominant blood type AB. A self cross between IB and IB in the F1 generation, the outcome in the F2 generation are, so the gametes are IB. Here also IB. When the IA gametes fuse, then the result is IA, that is blood type A or blood group A. Here the gamete IA and the gamete IB fuse, and the result is IB, that is blood group AB or blood type AB. Here also IA gamete and IB gamete fuse and form IB. This is also blood type AB. Here IB and IB. So the gametes fuse and form blood type B or blood group B. So we have one IA, that means blood type A. 2 IB means blood type AB. Here 1 IB means blood type B. So the phenotypic and genotypic ratios are same, 1:2:1. So this is another example for the codominance. Codominance in skin means hair color of cattle. So let us observe. The codominance occurs when the both traits appear in a heterozygous offspring. For example, Royan shorthorn cattle have codominant genes, means complementary genes for hair color. The coat means skin has both red and white hairs, not pink hairs. Here both red and white hairs, the both colors appear in the phenotype of F1 generation because of the dominant genes. So red color hair also they are also having the dominant genes, and also white color hair is also having dominant genes. So they fuse and form the both color appearance of the both colors in the F1 generation. That is a new character which is represented by the dominant genes which are present in red and white color hairs. Okay, next slide. Codominance in combs and fowls. While studying the combs of fowls, Bateson and Punnett scientists noticed a very interesting phenomenon. Wyandotte fowl breed carries a papillate comb known as rose comb, while Brahmos fowl breed possesses, which means has a high and narrow comb known as pecomb. Means Wyandotte fowl has rose comb, Brahmos comb has pecomb. Leghorn and such other breeds of single variety. So single variety comb is present in Leghorn and other fowls. Okay, next. Okay, if we observe the codominance in combs and fowls, if any one of them, means rose comb fowl or pecomb fowl, is crossed with a single comb fowl, the ratio is 3:1. So 3 rose or 3 p comb fowls and one single variety comb fowl are obtained in the F2 generation. When rose comb fowl was crossed with the pecomb fowl, an entirely new type, namely walnut comb, appeared in the F1 generation because of the dominant genes are present in rose comb fowl and also pecomb fowl. So they are complementary genes, they complement each other to express a new character, that is walnut comb. So the walnut comb is a characteristic of Malaysian breed. Okay, next slide. In this slide, the phenotypic ratio of dibate cross. If a cross is happened between rose comb fowl and pecomb fowl, the ratio is 9:3:3:1 in the F2 generation. So the rose comb fowl, the capital R, small p, and the pecomb fowl, smaller capital P. So a cross between these both, and the result in the F2 generation 9 walnut comb fowls. Walnut comb fowl, which is represented by capital R, capital P, capital R, capital P, small p, capital R, small r, capital P, capital R, small r, capital P, small p. If you draw the Punnett board, you can get all these 9 walnut combs, just you have to practice. Rose comb 3, represented by capital R, small p, capital R, small p, and P comb 3, these are represented by small r, capital P, small r, capital P, small p. Single variety comb is represent only one, represented by small r, small p, small p. So just observe this. Here capital R, capital P, both are dominant alleles. If they are present in the combination of these, a new variety walnut comb is represent. Any one of them is recessive. If the P allele is recessive, then rose comb is appeared. If the R gene which is recessive, P comb is appeared. But if both the genes are the alleles dominant and fuse and form a new variety comb, that is walnut comb, which is present in Malaysian breed. So this is about the codominance. In our next class, we'll observe the next new concept. Thank you one and all.